So I'm going to mute you guys, and then I'm going to press record, and we'll do the introduction. Cool. Well, welcome, guys. My name is Chad Bertram. I'm the Diving Safety Officer at the Audubon Aquarium of the Americas, and today we have Bevlina. Did I say it right this time?
Yes.
Yeah, I've only known her for like 10 years, and I finally said her name properly. She is an assistant professor at Iowa State University. I met her in Catalina while she was doing research working with Bluebanded Gobies. And I had no idea what she was doing then, and now she's here to talk about it. And I still don't know what she's doing with them. So we're really, really excited to have her. So thank you so much for being here and being a part of this.
Hey, thanks for inviting me. And, you know, it's definitely always special talking about gobies and then, you know, reminiscing, going to Catalina, because of course I'm not there this year. I had planned to be there already, but things did not quite go as planned. And yeah, so instead I get to talk to you guys in New Orleans. And so it's interesting because if you see the picture back there, you can't really see it well, but I actually bought it when I was in New Orleans 4 years ago at a conference. And it's about like, it's basically a cartoon of, you know, people playing instruments, and that was my memory of you know, wandering around French Quarter and all that. It was beautiful. So it's truly a pleasure to be here and share a little bit of what I've been doing for the past few years. And I will begin to share my screen, so hopefully it works uninterrupted. There we go. Okay, so this is basically a glimpse of what things look like in the Pacific Ocean. And you can spot a few creatures here, I'm sure. So today I will be talking to you about diving in Catalina, off the coast of Catalina Island, and what kind of research I do. And it's mostly not, it's not quite that I, you know, do science diving, but I dive to collect these beautiful iridescent gobies, and then bring them up to the lab. So in future, hopefully I'll be able to go out there and do some more field-based research. But they have so many predators around, if you can notice, that they don't quite want to come out and show their whole repertoire of behaviors, which is why it's just easier to take them in the lab setting. So first I wanted to start by, you know, saying thank you to the funding agencies. Like I started my research in Catalina. My first summer was 2009. And then that was as a graduate student, when I had just started my PhD program as a first-year graduate student. And then 2019 was my, like last year was my first field season actually leading the team. So it was definitely a very interesting transition. And this was, of course, it was a much larger team. There were a couple of graduate students and a couple of collaborators. This is my PhD advisor, Matthew Grover, and he was extremely kind that he came again, it was last year, to help us out. And this is really the scene that you would see at Wrigley when you're not facing the water. But I will definitely show you more photos today when you face the water. And now my lab is located at Idaho State University in this building here. And there are so many hills surrounding this town that without the water, it's almost like living in Catalina. So here we have sagebrush instead of palm trees. But it's a very desert scrub kind of habitat. Like if you notice these hills, it's brown and dry in the peak of summer. So yeah, and we've had a lot of undergraduate students helping us with this work. Typically, typically we go out with at least 2 or 3 undergraduate students, and I think that they get a lot off from this experience. So I wanted to give you a general overview of my research. And what I basically study is how the environment, which it could be seasonal changes, so for animals that live in temperate regions, such as where we live, they go through seasonal changes in a lot of things about the way they live. Like for example, one of the things you might be noticing now is that you can hear birds chirping and a lot of activity. I mean, you're more south, so I guess you're closer to the tropics, but generally the more north you go, you've got more activity for breeding season. The summer is generally considered as a breeding season in non-tropical areas, but in tropical areas, they kind of breed year round. Okay, so those are changes in how there could be climatic changes or these seasonal changes that occur, and then that leads to changes in the way animals behave. Okay, they have to get ready to behave in a certain way, and the way they get ready is by one of the things that kicks in internally is hormones in their body. Okay, so the climatic changes or changes in seasons also feed back on how these animals that are social, they're like, for example, if we go on with the example of birds, they establish their territories and they sing around their territories. So the way they sing will affect a neighboring bird, and they may establish that this is my area or this is my mate. So that is a behavioral consequence. And so these factors can affect the physiology. So what we mean by physiology generally is heart rate or breathing or your neuromuscular system or how your metabolism is. And these are words that hopefully you've heard of these words in the past. So I don't want to use too sciencey words, but I want to try to break it down a little bit. But what's interesting is that for all of these conditions, like all of these physiological aspects to run smoothly, there's often hormones are required for these processes. And there are different hormones that can affect different parts of your body, okay? Or the same hormone can affect different parts of your body in different ways, okay? And then morphology means what a part of your body looks like. Like, morph means the way a part of the body looks. So hormones can often influence what a part of your body looks like. And all of these together basically affect the ability of an animal to reproduce. And the whole point is to pass on genes. Okay, so down here, I basically have all of these different species that I work on and I've worked on in the past. These are white-crowned sparrows, and these guys migrate to Alaska to breed. So I had, for my postdoctoral work, which was at UCLA, I had worked on, what kind of hormones change in the muscle when these birds are migrating, okay? And then there are these birds called the golden collared manakins, and they live in Panama, and they show really cool courtship behaviors. And so they show these plyotropic jumps, like if you've heard of what box jumps are, these birds do that to attract their mates. Okay, and then these are song sparrows, and I studied these guys when I lived in British Columbia, and I worked on their aggression in the non-breeding season. And these are zebra finches that live in Australia. But in neuroscience research, a lot of people study them because they're actually very easy to maintain in a lab. So the reason I told you about all these birds is because they're very much a part of who I am. But then I also have been working on fish. Most recently, I've been working on these trout, which are red band trouts. They're kind of like rainbow trouts. They are a variant of rainbow trout. And they live, I mean, rainbow trout live almost, I think they're pretty, they span a lot of different river systems in the US. But these guys, the red band trout, are most prominent in Idaho. So I've just started working on I got a regional grant to study stress physiology and seasonal impacts on red band trout. But today I will be focusing on these guys, which are the blue-banded gobies. And these guys are quite a bit smaller than trout. Like a really big goby is the size of your pinky. Okay, so that's really how big they are for reference. So, which makes them kind of, I don't know, it's like it's kind of easy to study because you can put a lot of them and manipulate them in smaller groups. But then when it comes time to study hormones, like all the parts of their body are really tiny, so it makes it a little bit difficult. But they're really fun, so that's why I study them. And so today I will start off with, what I'm interested in with hormones. And then that might be like the most sciencey part. And then we're going to go into, what diving in California is like. And then we'll go more into hearing about these gobies. We'll just transition into what blue-banded gobies are and what is their life like, and then a little story about integrating the hormone and in the life of blue-banded gobies. Do you have any questions so far? Or okay, we're just gonna go ahead then. So when I talk about hormones, like most specifically, I talk about steroids. And you may or may not have heard of steroids in the past. But steroids are naturally produced in your body, okay? And steroids basically come from cholesterol. So this is what cholesterol looks like. And it has this long structure, which is basically called as a side arm. And that side or arm can be cut in different ways, and then you can get different kinds of steroid hormones. Okay, so there are, I hope that you may have heard about some steroid hormones, and we'll be talking more about those right here. The common ones, one of them is glucocorticoid. So glucocorticoids, for example, cortisone or prednisone. You can definitely purchase cortisone off the counter if you have any kind of rash or allergic reactions. You can also get nasal sprays, and that contains cortisol. So you can buy those for any kind of treatment where doctors don't have a proper a way to control like disease, for example, cancer or certain kinds of immunosuppressant or autoimmune disorders. Doctors often prescribe prednisone, okay? And so it's very common. Like when people say, "Oh, I'm on steroids. I've been gaining weight," it's usually that they're on prednisone. Okay, and there is a connection with gaining weight when you're taking these kinds of steroids that are termed as glucocorticoids, and it's a mouthful, but gluco comes from glucose. Do you have a question there? Yeah, you can just maybe I can unmute you. No, I can't unmute you. Chad, can you unmute her?
Okay.
Yes. Oh, you're gone again.
Oh.
Yes.
Okay. Prednisone, that's used, the reason I ask is we had a cat who got on lymphoma, and I think part of her treatment had to do with prednisone, because the idea was we needed her to gain weight and to be able to eat and feel good, and that was very critical to the treatment of lymphoma with animals. Like, yes, they're not going to live forever, but it does allow their chemotherapy to work. And it's the same one, right?
Yes, exactly. It is given like any kind of immunosuppressive disorder like cancer. Like that is a type of cancer that you just mentioned.
Okay.
Their patients are given prednisone.
Okay.
Yeah. So yes, that is probably the same one. There's also prednisone, which is also a steroid, but it's got a different kind of function.
Okay. I was just curious because it just seemed like maybe, that because it's hormones, and I figure cats or dogs, whatever, dealing with that.
And a lot of all we know about hormones really comes from other animals. And so if things work in animals, and especially in mammals, then like cats and dogs, then we end up using it as well. Yes.
Right.
Yeah, so great question. Please keep those questions coming. So like glucocorticoids, we naturally have glucocorticoids, and prednisone is a synthetic version, okay, that you can just purchase, that is produced commercially, and then we can purchase it. And what glucocorticoids do is that they mobilize our energy reserves, and yes, like we end up gaining weight as a side effect, okay. But it also is good for a variety of other things. Like it helps maintain our water balance in our body. It inhibits inflammation and swelling, and it's also actually important for the natural maturation of organs. For example, during the childbirthing process, if you go with a natural delivery, there's a huge surge of cortisol, which actually helps the child to transition from that environment when it was, in utero, and now it's, having to breathe air. So there's a big difference in how so they really need, like cortisol can be a good thing for several reasons. However, like it's got a negative context or negative connotation, because one of the things that is used to understand how cortisol, or how you might be doing, if those people who have hypertension, for example, they go through a stress test, okay, and the stress test involves testing cortisol levels. And so usually it involves salivary samples, so you can spit in a tube, and then they can extract cortisol from that. Cortisol, of course, it's involved in regulating blood sugar because of that glucose thing I was talking about. And then actually goes down at night when you sleep. And then first thing in the morning when you wake up, you have a burst in cortisol, which tells you, okay, get up, go eat breakfast, and so forth. And then it starts decreasing again throughout the day.
I have another question about that.
Yeah.
You know how they talk about how as people get older, or women dealing with menopause and all that. Is it that the cortisol happens earlier? You think that's why they have that thing of waking up early, and they always talk about that with it, that
That's
you're.
a good question. Actually, like our brain, so waking up, like our sleep and wake cycle, all of this, like we have centers in our brain, in particular regions of our brain, and our brain is filled with hormone receptors.
Right.
This is a great question because it'll be really helpful for you later to.
Okay. Well, I'll shut up then. I was just curious
but
because
it's true.
I thought maybe that was it, that a lot of people complain. And if it's happening, it's something that you really have, I mean, you have to adjust for that or something so that you're not waking up like 2 or 3 hours before you really normally would have 10 years ago. I do that right now.
Well,
so I'm just.
Sleep is actually, like, we don't understand sleep physiology yet.
Mm-hmm.
And there are so many different factors affecting sleep. So, so I mean, there are the cortisol, like there's melatonin, melatonin that you can purchase over the counter. All of those things can interfere with sleep.
Yeah, with the balance of when it happens.
Yes. Yeah. So your body's internal clock, it's very easy to mess with it.
Mm-hmm. Yeah. Yeah, it's
and these hormones are all connected, and so you've got your question about menopause brings me to sex hormones, because that's really the other thing that I study. And so when people have heard about sex hormones in the context of being anabolic steroids. Okay, but then a lot of times, so there was also this thing called, for menopausal women, there had been a huge study on hormone replacement therapy. And there are many kinds of hormone replacement therapies, and one of them could be using this estradiol cream that is prescribed, that is premenopausal, when women start showing symptoms. Okay, so these sex hormones are really important for reproductive behaviors and how we, interact with the opposite sex, and also our also turning on secondary sexual characteristics. Okay, so this is, the wattles and combs of roosters is the most prominent example in among scientists, when it was first discovered that these hormones are influencing these kinds of characteristics that these hens find attractive. And so these hormones are so testosterone, we generally think that it's produced by the testes, and estradiol is produced by the ovary, but really both men and women have both of these hormones. Okay, and it's mostly because testosterone actually can be converted to estrogen. So we don't have one or the other. We have both of these, but the amounts that we have is different. Okay. And then your cortisol is generally produced by the adrenal glands. Okay. And in fish, which I'll be talking about today, they don't have, there are different kinds of locations of adrenal glands. They're called adrenals because it means on top of the kidney. But fish generally have these glands in between kidneys, so they're called interrenal glands. Okay, so if you go, generally when we get, when our doctor wants us to measure hormones, we usually get a blood test, right? So that is so all of the red structures here are hormones, and usually these steroid hormones, they cannot just be free floating in the blood. They're actually bound to other proteins that help them get to their target, or also prevent premature actions of these hormones. And so, the most common route that we humans are used to is taking samples from the blood. But then there could be other ways. Okay, so for example, I was talking about the topical estrogen kind of, you can have patches, or you can put the cream. And that's because your skin is actually absorbent to the hormones, to steroids. And then your hair, or in case of birds, feather, or fur, like, in bears, you can actually measure hormones from that. So it's kind of an indirect measure of hormones rather than, going for the blood. I think people would think twice before chasing bears around to get to get hormones from them. Another way is saliva, which I just told you. So you can just spit in a container and we can extract the hormones from there. In the cattle industry, they use urine. Okay, they collect buckets and buckets of urine to test for whether or not those cattle have been treated with antibiotics or hormones. And you usually see those labels when you purchase, I don't know, yogurt or milk or whatever. And that's because they, dairies, they have to report these things, and they usually use urine tests. Okay, which is same as, a pregnancy test. You can measure, and that's how you can do measure hormones. There's also other indirect ways that, like, for example, you can measure hormones in poop also. Okay, and that brings me to, if you can recognize, our Blue-banded Gobies. These guys are so tiny that they don't have that much hormone that you can measure from blood. So what we do is that, if we have to do that, yeah, we have to kill them. So we cannot do repeated measures on the same fish. And then too, we may not get enough that's required to be detectable. So what we do is another indirect method is that we can actually put them in a cup of water, and then we leave them in there for an hour, and then we just measure how much hormone has been transferred or exuded into that cup, into that water. So then we can extract that and we can measure that. So these are kind of all these indirect ways. A more direct way could be like actually taking a biopsy. Okay, so for example, to detect certain kind of cancers, you do a biopsy. And so that's the next level, like, getting closer to the tissue, or, or doing, or getting closer to the source of the problem, right? Or you can measure all these other things like receptors. We don't need to go into detail, but you can measure enzymes and receptors and all kinds of other things, which we usually do in our lab. So our brain also produces hormones. And so I told you earlier with the sleep thing that, there are glucocorticoid, there are receptors, like these molecules that can detect cortisol in our brain, or melatonin in our brain, that regulate our sleep-wake cycle. And so basically there are different structures in our brain, different nerve cells that can detect hormones that are coming from the rest of your body, but your brain can also produce those hormones, which is why because I'm really interested in the brain and how the brain regulates our behavior, that's why I generally study hormones in the brain. And I can do that, relatively easily with these gobies. So, yeah. So, and I wanted to talk a little bit about another misconception that people might have, and that is like, just because you have a lot of testosterone makes you very you know, makes you a very angry person, very volatile, and you can, you know, you respond to almost anything. And you know, it's mostly, it's what we talk about context. Like, it's not that people with high testosterone are constantly angry and getting into fights, you know, but maybe that they, based on, you know, what kind of environment they're in, could affect their propensity to get angrier quicker, maybe. But it doesn't really work that way. Just because you have high hormones does not mean that you're going to be like that. Do you have a question there? Yeah, go ahead.
Have you ever run across something called dehydroepiandrosterone?
Yes.
DHA?
Yes,
I
that
mean,
was
it
the
may not have.
first hormone I ever studied, and I've
Really?
got several papers on that. Yes.
Well, isn't that interesting? Does it? I mean, I don't want to derail anything. I was just curious. Does it affect any of your research with the fish, or is this just something that's because of what you do? You did study this, and it. I'm just curious what you thought of that.
Okay.
Hormone.
Well, so what dehydroepiandrosterone is, or what we will call for short, is DHEA. DHEA is considered as a precursor. So it is a chemical from, like, you know, remember that image of cholesterol? When it's cleaved up, it's cut, you know, that side chain is cut up in different forms. And when DHEA is considered as a prohormone. So testosterone is actually produced from DHEA. And DHEA by itself is not very potent, because it won't affect you the same way as if you pop testosterone pills.
Sure.
Because it does not have a receptor in your body that it can bind to.
Okay.
Yeah. However, what DHEA can, like, what can happen is that DHEA, if you take DHEA, it can be converted to testosterone and then eventually to estrogens. So it can affect you, like, in that way.
Right. Okay. And
Yeah.
I heard that it had something to do with that people, like, when people have taken it, that it actually helps maintain or at least keep a person fertile, because of the fact that it kind of lengthens that period of life, whereas
so what happens, what happens in humans is that DHEA is really high just when you're born, and then it just plummets. It's like absolutely, there's hardly anything there until you hit puberty.
And then it's
So
up.
then it increases, and then it starts decreasing for the rest of your life.
Yeah.
Which is how you've got, you know, that's how you've got all these other hormones also decreasing.
Right. They're all interacting with it. Yeah. Yeah.
All kinds of. Yeah. And so again, it shouldn't, in my opinion, in my humble opinion, it shouldn't be allowed to be purchased over the counter. But you can purchase it over the counter.
Right.
And it can actually lead to very similar effects as, you know, taking testosterone.
Sure. Yeah. Yeah. It can have an effect on women, especially. Yeah. Okay.
Yes.
Well, I was just curious. That's interesting that you did. I just have a.
Yeah, and I actually worked at. Yeah, and I mostly worked on that hormone with birds. I haven't yet looked at them in fish. So I do plan on it in future. It just, I'm just starting, you know, it just depends on which students I get and what they end up doing. But yeah, I would love to look at them in fish. But I've asked a lot of those questions in birds, and I understand it quite well.
Very interesting. I didn't know birds would have, I mean, you know, I guess, since they're not mammals, you wouldn't think.
Actually, that's what's interesting about these hormones, is that all vertebrate animals, amphibians, snakes, whatever, you know, they all have hormones. They may have different types of hormones that are more active or not, but really, like, that's really the reason that I study other species, is that we can be pretty invasive with them. We cannot do the same thing with humans, but we can actually study how these hormones function by looking at this in other species.
Right. Cool. Okay. Thank you. Yeah.
So so high levels of testosterone does not mean high rates of behavior, and so this is again like this was shown in a classic experiment that was published in 1949 by Frank Beach and Marie Holtz Tucker, and I couldn't find Marie Holtz Tucker's photo because I guess when you were a woman in that time, you're not really, you're kind of just doing things in the background. But basically what they had is that they had these rats, okay? Or not rats, I think they were mice. Anyway, they were some kind of rodent. I'm not good with my rodents because they come in so many different ways. But this would basically work with rats too. So you have these rats and they measured their level of sexual behavior. And what they found is that you can put them in a scale, okay? So they can range from very low levels of sexual behavior to very high levels of sexual behavior. Like if you give them a mate, they want to copulate that mate constantly. So they show a very high level of sexual behavior, but they are all in a range, okay? So it's kind of like a normal curve, like it's like height and weight. If you graph people's height and weight, it forms into a normal bell-shaped curve, and so that's how this is as well. Okay, so the idea was, okay, in baseline, you've got all of these males with high levels of or low levels of sexual behavior. What happens? Like the question was about testosterone. Okay, so do high levels of testosterone make you more, behave in a more have higher rates of these sexual behaviors? So what they did, like endocrinologists, that's what people who study hormones, what they tend to do, there are very few types of experiments. They either give too much or they take it away. So in this experiment, they took it away. So what they took away was the source of the hormones. So what we consider is their testes. Okay, so they castrated these mice, and they basically had no more testosterone. Okay, and their levels of sexual behavior also became zero. Okay, so they would not take away the testosterone, they will not be sexually active. But then what they did is that they gave everybody low levels of testosterone. All of these rats, no matter what, or mice, whatever, no matter what level they had before, they gave them all a very low level. But what was interesting is that no matter how, like even though they gave them all the exact same amount of testosterone, they came up with that distribution where they had high or low levels of behavior. And it was those animals that had high levels still had high levels, even though they had the same levels of testosterone as these guys. So that basically shows that just because you have high levels of testosterone in your blood does not mean that you're going to behave, with the same increment. Okay. So, the other thing that like a lot of people who are, my friends who have been becoming parents recently, they're worried about, how does testosterone affect parenting? Okay, so like males who are parents, how are they going to behave with the child? And there have been some studies, and somehow all these studies are in Filipino men, and all of these studies look at when once men get married and then once they have kids, they have lower testosterone levels. Okay, this is found in other, in these are juncos. So these birds also, like, they have biparental care, and so males have lower testosterone when they're dads. That's interesting. But that's, you know, measuring testosterone in their blood or their saliva. There's also these monkeys that have lower levels of cortisol. So corticosterone in monkeys, which is kind of the same as cortisol, but they have, so I mean, whether you have high levels or low levels, it's still not telling you about the function really, okay? And there's kind of a mix in literature about what that really means. Okay, and in case you were curious, this is my dad and this is me when I was younger, quite a bit younger. Um, and so there are these other, you know, mammals. These guys are desert mice, and they have biparental care as well. And what we find in them is that dads actually have higher levels of testosterone. Okay. So, and then we've got these fish, our blue-banded gobies, and they have a version of testosterone that's more active called ketotestosterone. And they also seem to have higher levels when they're parenting. Okay. So it's actually the dads that take care of the babies, which is what I will be telling you a lot more about next. Okay. So now I've kind of taken a while, but I've, you know, I've gone through the hormone part, and now I'll be segueing into, you know, I've got Chad smiling there, takes him home again to Catalina, I think. So this is a lab area of USC's Wrigley Institute, and they have really great facilities for us to do our research. This is kind of what the morning looks when you've got all these people like getting ready with their dive gear, and this is where it kind of leads into the locker room on the other side. They have like gas filling stations here. And then this is, of course, when a little bit later in the day when there's no one around, it can be really calm and it can be actually very relaxing. I love spending time on this dock, just laying there. And this is, we have access to boats. So we use boats and that we take out into the ocean, and I'm suspecting that it's very different, like when you dive where you are compared to when we dive in Catalina, because you have to be suited up completely, and you can get it. It can be very cold, and so we have not just, you know, all this bulky, all of this stuff that we are wearing, but then also for, you know, usually we collect fish, so we have to have all our gear, and we have to make sure that we come back with our fish, otherwise that whole dive was useless. So we usually have a lot of hooks and all that attached to our BCDs and all kinds of things. So yeah. And this is just a like pretty picture of Catalina at sunset, you know, and there's all kinds of things going on. It's never a dull day. We have, you know, visitors often, and they'd actually taken bison over to, in the 60s to shoot a film. And so they have, I don't know, hundreds and hundreds of bison moving around. They're not supposed to be in small islands like Catalina, but they have so many bison there. And what happened is that in the end, that movie, they actually ended up leaving them there because it was too expensive to take them back. And also, they actually cut all of the parts of the movie that had these bison in them. So we have some big mammals around. They come into, like, this is one of the places in a town called Two Harbors where we stay. Like Wrigley has these houses for us that we, you know, can pay rent. So, but it's not too shabby. We get to see, this was, you know, Trevor had a lot of fun trying to get this guy out of his, this is his skiff. remember that day I'd taken a lot of pictures of this guy, and this is a nightingale, and we can hear them every evening. So yeah, and these are the different places that we collect fish from. So there are reefs that that contain lots of sea urchin, mostly around these different areas: Bird Rock and Isthmus Reef, and then there's Shiprock. There are other places too, but these are the places that we tend to go when we take our boat. And so we just load up everything, and then we get to these places, we anchor in, and then we are in the water. And these are just some glimpses of what things look like. So you have to go, like sometimes 45 feet under. And there are really these walls of coral that have sea urchin, and we inject some anesthetic into the ocean in small places, and then we swoop in these fish, and you can get some context for how big they are. And then we can collect them all in this jar, and then we have to make sure we bring the jar back, and that can be a problem sometimes. You have to, everything is very orchestrated and has to occur at specific times. And and then my first summer that I was diving, I was not very good at keeping track of these things, and so my job was mostly like, I had one of these hand nets in with me, and then with the other hand I was actually punching sheepheads or Garibaldi that were could come and eat our gobies before we got to them. So that was kind of my job, was to keep the predators away. So it was interesting. It all has its own beauty, and I can, these are juvenile Garibaldi, and they have a sculpin. I think that last summer one of our assistants actually shot the sculpin and then brought it to the dinner table. And then we get to see all kinds of things. These guys are my favorite, the nudibranchs. And sometimes we have bycatch that we bring into the lab. So this is an abalone shell, and you can see the underside. This is actually filled with eggs from these gobies. Okay, so these orange eggs are like, these dots are all orange eggs, and you can see that with the naked eye. And these are all newly laid eggs, whereas all of the black dots, those are also eggs, and those are actually eyes of the eggs. So as the eggs are fertilized and they start developing, you can actually see the embryo move around, and the first thing that forms are the eyes that you can see, and you can track them. So this is a bucket in which we put all of these fish after the dive, and then we often put the fish in the community tank, and then we have to bring them out and process them under the microscope to measure them. These are we have indoor facilities where we study fish in small groups, or we can actually put them outside if we want that. And this is again my PhD advisor. My first field season, he was teaching my lab mate Tessa, and all of us really, how to measure the fish and things. This was actually, I'd gone over there to do some outreach work. So Wrigley Institute is really big on public education and engagement, science engagement activities. So I had been invited to go teach some high schoolers about these gobies. So that was fun. So I'll get to teach you about these gobies now. So we're kind of moving on from talking about, the environment they live in, to, what makes up the life of these fish. Okay, and they are very interesting because they live in small social groups. So like it's kind of like a family group. And what we do in the lab is that we assign those families. So we measure them, and then we look at some features that can help us identify whether they're males or females. And then we can put them in their groups, and we give them a little PVC tube, which usually forms their nesting site. And the male is the one that is the most dominant in that group, although he's not very aggressive. It's a female, it's mostly the alpha female who's most aggressive. Like she's always going in, kind of in attack mode and approaching all of the other females. Okay, so I'm going to show you a video of one of these groups. Okay, so there is a male inside. Sorry about the quality. You can actually see the reflection of the glass with the hill, the back and things, but try not to pay attention to that. So we'll start off with this male who's inside the tube, and there's a female here on top. And what you'll see is that you'll see the male fanning and rubbing eggs that are inside the tube. Okay, so there we go. So here he is. He uses his fins to kind of rub these eggs. And then he comes out, and then he chases females away. And he goes back in. And then this is kind of, it looks like a chase, but the way he was moving was, it's called it's a jerk movement, where he moves as if he's electrocuted. And that's actually considered as a courtship ritual, where he indicates that he's ready for the female to come inside and lay eggs. And then the females are also constantly like approaching and displacing each other, and that's how we measure aggression. So I'm going to show you this video one more time without me talking, so that you can actually pay attention to what I was saying. Okay. Yeah, so I mean, these fish are really easy to watch once you learn what these different behaviors are like. We usually watch them in like 10-minute intervals, so it's kind of almost like watching a mini movie, because you're like, oh, like who's who's beating up whom and things like that. It's kind of fun to watch them, and so it gets easy to train undergraduate students as well to watch them. So yeah, high schoolers can count behavior. So it's oops, I'm gonna proceed to the next slide. Okay, so what we do is that, like, once we've got these fish, we'll put them in groups, and so we'll just sort them by size, and we have them 3 millimeters apart, and you can see that this one looks the biggest, and then we have the rest of them are all like smaller. And then if you wait for a couple days, they actually establish a hierarchy. So you can actually tell very easily that they sort them up. So they sort each other. They're very civilized. They know how to sort themselves out, more so than, than others, and so once they have their position set, unless there's a disturbance, they basically keep that hierarchy. Okay. And we kind of choose that, we predetermine this 3 millimeters difference because it helps us to, it kind of just helps us to ensure that this hierarchy actually stays stable. If they're all very close in size, then it might just take longer for them to resolve. Okay. So one of the things like we use, you know, to tell them apart is their feature of, you know, males and females. And so we tend to measure their dorsal fin, and that's something that we only recently started to do. And so we found very recently that, you know, males in general have much longer dorsal fins than females do. You could have really big females that have bigger dorsal fins, but we've not yet figured that out. But for the most part, we've done this repeatedly with like 100 fish, and we find that for these 100 fish, 100 males will have larger dorsal fins than 100 female fish. And then this is a juvenile fish in yellow, and they don't really have that difference, but you know, there's could be a slight difference compared to females, but we didn't find that to be a big, you know, big trendsetter. And just so that you can see this more easily, like these are there's a male and a female in here, and all of these are orange eggs. And so these orange eggs, they adhere to the inside of these tubes. And so that's how the male basically is inside that tube all the time. And what we tend to do is that we can slide in a piece of acetate. So like the transparency that our professors used to write in the olden times, we actually use those to line the tubes, and then we can actually pull them out and we can photograph those images to see how many eggs were laid. Okay. And then the other feature we use under the microscope is looking at their genitalia. So they have this organ that the male uses to transfer sperm, and then females use to deposit eggs. And this is a male here, like it kind of looks long. So it's long and skinny in males, and it's more rounded looking in females. And so we can, that's how we sex them. And what happens when they change their sex is that it'll go to a more intermediate stage. So we'll talk about sex change later, but we can actually induce sex change by giving these fish hormones. You can give a female ketotestosterone, which is the fish version of it, you know, of the more like of testosterone kind of. These guys have testosterone too, but you can give a female the male hormone and she will start having male-like genitalia. Okay, and then so this is what the females look like if you cut them. So if you cut them out, like you can see these eggs. They kind of look like, you know, if you eat sushi, it kind of looks like that. All the eggs that you eat, sushi. And then if you cut them in cross section, so if you cut them like, you know, midway like that, you can actually see these are all lipid droplets, so fat, like containing yolk. And then this is a male testes, and they're filled with sperm. So they stain very differently if you put different colored dyes on them. Like you can make very thin sections, and then you can put different dyes. And the male testes look like spaghetti. Very interesting. But then they also have this structure, this thing here. This is called an accessory gonadal structure. And this is very similar to the prostate gland in humans. Okay, so yeah. So one thing that like my lab mate, Tessa, she had pioneered is basically she injected these fish with different kinds of food coloring, and she could make eggs appear as different colors. So you can see the orange and the yellow and the green. And then we can see, you know, which fish may have laid which egg. So it was really fun. Okay, and yeah, and so like after the fish, you know, after they hatch, usually like they're really tiny and transparent. These larvae usually like to drift in the plankton. And then as they drift out, they get bigger, they look quite transparent, and they don't have the coloration until they start settling onto the reef. And once they settle onto the reef, they join these groups. So I was talking about the sea urchins. So usually every family group in the ocean, like they're associated with the sea urchin. And you can see that, you know, there'll usually be a male and several females. So as I said before, they're polygamous. And the males, you can tell that he's a male often like by looking at his behavior, but you can't be confident until you put him under the microscope and look at his genitalia, whether or not he's a male. And then you've got these females who have a, you know, a hierarchy, and then they have these specific types of displays where when they get gravid with, you know, when they're filled with eggs, they kind of show off their belly to the male to, you know, want to go and lay eggs for him. They also, you know, show adult sex change, which is really interesting, and I'll be talking a little bit about that. It looks like it's past 7. I think you'll go on for a few more minutes, so hopefully you're not, or it's past 8 o'clock for you guys. And so they also have these guys called mini males who pretend to be females, we think, and they kind of can steal eggs, you know, so that their sperm fertilizes the eggs. But then the male, who's, you know, in charge of that territory, he is still taking care of those eggs. Okay. And so this is kind of like they're spawning, they're like inside the tube. And this is this male in this shell that never got out for the whole summer. So he actually laid eggs in there. And I mean, he laid his sperm there. He was caring for those eggs, and you know, you can actually see these eggs quite well. So it was just fun, and we can count those eggs to see how successful they are at breeding. So I've been talking about sex change a lot, and I kind of held like last thing I talked about was the stable group. So you know, after 1 to 3 days, depending on the group size, they're considered stable. And what we can do to disrupt their hierarchy is we often remove the male. Okay, so when we remove the male, we have all kinds of things happening. Okay, and it's very interesting to see what's happening, but usually it's the most dominant female who will then take up the nesting site and be transformed completely into the male. Okay, so all her structures inside her body will change and she'll become the male. Okay. Oops. So it's kind of like when we study sex change. I stole this slide from my PhD advisor, but I really liked it. So basically, when we study sex change, there's this thing like that we look at as the like the behavior of that animal, that we can tell whether they're going to be behaviorally male or female, because you know those males have very specific, you know, they're attached to their tube and they're defending their eggs and all of that. And then we can actually measure their hormones that can tell us whether they're male or female. I told you that you can actually cut them open and you can look at their gonads, you know, to see whether they have eggs or they have testes. You can, you know, externally, without having to cut them open, you can just look at their genitalia. You can tell whether they're male or female. And then, you know, when they're male, like, you know, female going to a male, they grow this prostate gland out of nothing. So it's like, we don't really know how that comes up all of a sudden, but that is also is really interesting. And they really have all these changes like all throughout their body. So they will get bigger over time. They not necessarily in these fish, but in other fish, they change color as well. And it's possible that these fish also change their fin size, and how long that takes, we don't know yet. My last few minutes, I wanted to like just put together some hormone stuff and some things that we know about, you know, the behavior of these fish. and things that we've discovered in the past few years. So, this is basically what it is like in a stable group. Like you've got the male and the alpha, they're spawning, they're happy. The male is caring for the eggs. The male keeps the female out of the nesting site. So, before I was starting to do my PhD, like my advisor was not really looking what was happening inside the tube. They would just look at, the process of sex change. But I was very interested in studying how this male, defends his eggs and what does he do? And, I started looking inside the tube. And so I spent hours and hours looking inside the tube. And when it got interesting was I wanted to find out what could we do so that the male is not in his tube. Okay, so what we can do is that we can actually inject the brain of this male with a drug, which is called CBX. Okay, I'll just call it CBX for short. And this drug actually prevents the male from entering his nest. So at least for like about 60 minutes, he does not enter his nest. So that means he's not caring for eggs. Okay, so what happened is that when the male was not inside the nest, we had females entering the nest. And this is a bit too complicated, but what really happened, like if you look at these dotted lines, it's actually like this increase of females eating eggs. Okay, so when the male was not taking care of the eggs, he's still around. He was actually interacting with them. He was outside, he was approaching and displacing them and all that, but he just wasn't entering. He wasn't associating the nest tube as something where he should be, which is interesting because usually, like when we try to take the nest tube out to take the eggs out, the acetate, the male does not want to leave the tube. He comes out with the tube. Like you have to push the male out of his tube. He does not want to leave that tube. And so here we altered their behavior where they were not entering their tube. They were hanging out outside. And meanwhile, we had these females who started eating eggs. And also like within, about an hour, so it was like different for different fish, but, max, like over an hour, basically, the male lost 15% of his eggs. So that's a lot of eggs very quickly. If you're not taking care of your offspring, they're gone. That's really the consequence of you not guarding your offspring. Okay. And so what this chemical is that it actually blocks ketotestosterone production. Okay, so we basically injected this drug that blocked all kinds of ketotestosterone in their brain, which stopped them from being parents. Okay, so that was really interesting. I mean, that was the highlight of my time. Oops, this kind of messed up my animation. That was the really fun part of my time in that lab. Because there was actually something, oops, I'm gonna go back. There was something else going on. When that male was not inside the tube taking care of his eggs, the female started eating those eggs. And actually the most dominant female, the alpha female, she actually started then showing parenting behavior. She started fanning and rubbing the eggs. Like she would take a nibble of the eggs and then she would also fan and rub the eggs. And that's something that we did not expect to find because it was so quick, just within a few minutes. Like this was time in minutes. So like often within 30 minutes, like you had females showing like egg care as well as like, they were eating eggs. It was weird, that transition, but they start transitioning to wanting to be inside that nest and being territorial. And so what we found is that those females who were behaving like males, like showing the nesting behavior, they actually had higher levels of ketotestosterone in their brain compared to females that were only eating eggs. So I hope that was not too complicated, but that's basically what I've got here is that, if we have this male that we, prevent him from either entering the nest or we take him out of the group completely, the most dominant female, she will transform to the male. And she starts exhibiting those parenting behaviors. And she actually has higher levels of ketotestosterone in her brain. And, with that study where we blocked ketotestosterone in males, we can tell that it's something to do with parenting. So there could actually be similar things going on during this change that's affecting their parenting behavior. So yeah, so if, just to wrap things up, it was so quick how they change their, how quickly they start showing their male typical behaviors. And then these females, we had never seen that before. Before I got to the lab, they weren't actually looking inside the tube to see what the females did. So they can actually increase their nest care behaviors right away. And then I think that there might be something to do though with how their movement is. So the males, when this is a male inside the tube and this is a female inside the tube, the male is a bit larger. His fins, if you notice, they're actually touching the eggs. And when they show their fanning and, fanning behaviors, they actually brush over the eggs physically. Whereas females, their fins are not long enough and they're also more retracted when they are in the nest. And so they're not using that dorsal fin a whole lot, but they're mostly using their pectoral fins. So I would really like to, apply for funding for future research where I look at how this fin growth is regulated and, whether there's something to do with hormones and how the brain kind of controls that, the movement of their fins. So yeah, that kind of brings me to the end of my talk, and I'm going to leave you with a photograph of Pocatello, where I live, and I'd be happy to take any more questions if you have any.
I have a two-part question for you, Devina.
Yeah.
Number one, have you seen any transitions happen without taking the male out? So, where they almost become hermaphroditic, like you would find in the California sheephead?
I'm not sure what exactly the whole breeding ecology of sheephead is, but I think for sheephead, they go through a transition, their lifespan is also a bit longer. So, and then the way they live, they're bigger fish, so they have larger areas that they go towards. So that's why we study a smaller fish that can change quicker too. Okay, so yeah, so they're a little bit different. So what they'll have really the only time that they can have another male is that if there's another male who's a mini male, okay, who's pretending to be a female, or if there's another nesting site, or that they start making a new nesting site close by. But otherwise, you cannot find two males, very close to each other,
Gotcha.
unless they have their own space.
Gotcha. That makes a lot of sense. And the other question for you is, how long of a transition is it from female to male? It's 24 hours or a little bit longer than that?
So yeah, so I guess I didn't touch on that. So when you have the transition, if you characterize that as a behavioral transition, it can be really quick, okay, within a few minutes or hours, and so that's one of the things that we, and then even in terms of their hormones, it can definitely happen within a few minutes and few hours, excuse me. So within 24 hours, they'll show differences in their hormones. In terms of like how quickly, like, they're plumbing internally and they're, they go from producing eggs to producing sperm. That is dependent more on, one of the ways that we track that is looking at whether there are eggs that have been fertilized. So if in the tube there are other, if you see the appearance of fertilized eggs, that means that that new male is now functionally reproducing as a male. And we've seen that in as few as 7 days, although it'll take more like 2 weeks for that to happen. But a female can reproduce as a male, like they don't do both at the same time, but they can reproduce as males even with 90% ovary. So they just shut off their ovary functions, but they start growing sperm. So as soon as they have sperm starting up, they can start reproducing. Yeah.
Gotcha. Great. Thank you very much.
Yeah, you're welcome.
Yeah, really liked the presentation. Very well set up,
you,
and I
Dan.
enjoyed the content you had. I had a question about, you mentioned about like the behavioral changes, but have you seen any changes as a result of climate change on the genetics of the gobies?
Okay. Well, I don't necessarily study that. There are other people studying those kinds of things, maybe. I think in future, if things continue this way, I might go study effects of climate change. I'm not sure if I'll study genetics right away, but I might study how they respond, because unfortunately what happened last year is that I lost a lot of fish due to this fungal outbreak. So they had this thing called ache, and they were coming, so in the water, in the ocean, like the ocean temperature was quite warm, and they were coming up with ache. And so when we put them in their tanks, and it's a flow through system, so all the water is kind of mixing in, that spread that infection. So even if they had just a little bit, like we would come back and like in 2 days, all of the fish would die. And that was really horrible. And initially everything was fine when the water temperature was cool. And then, after 2 weeks, when we went into July and it started warming up, and these fish started to have ache. And so I should have, that last year I didn't do this, but I think next time I go, and if these kinds of things happen, then I'm going to start following that a little bit more and seeing if I can, if this is really a thing, because it was just happening so fast that we couldn't really get anything more out of it than that, Yeah.
Yeah, I do have a question.
Yeah.
You mentioned that when you disable the male's instinct to be a parent, the females may start eating the egg. Is that because without that fanning mechanism, the eggs suddenly become inert, if you want to call it that, and they just sort of become food? Or is it that the females are doing it on purpose?
Okay,
if there is such a thing.
they will eat. So there are a couple things in your question, so I'm going to try to remember everything to address it all. But so the females will eat the eggs for extra protein if they can find it. Okay, so whether or not the males have been fanning, like even for a couple minutes, if they're, like the male, like one of the things he's always defending the eggs from is the females eating them. So it doesn't matter whether he's fat. I mean, yeah, it's just extra protein. Or maybe like some people have asked me, do you know whether they're eating their own eggs or another female's eggs? I don't know the answer to that question yet. So they could be doing that to prevent the success of other females, but they're, getting their genes through. I don't know that. And so those are interesting things to pursue. But the male, like what's very interesting is that whether or not he's in the net, whether or not he has eggs in the tube, he actually shows those fanning and rubbing behaviors. He's just so like, that's kind of mechanical. If he's in the tube, he'll spend 90% of the time that he's in the tube fanning and rubbing on inside the tube, whether or not there are eggs. And there's a group of Italian scientists that had looked at a different kind of goby, and what they found is that those gobies actually lay sperm trails inside the nest, so they're always ready if there are eggs laid. Okay, so they're always laying sperm inside the egg, inside that nest. And then the other thing that they might be doing is they might be spraying antibiotics, all kinds of things that keep that nesting environment clean, and ready to support growth of these embryos when it's ready. So they're always working. They actually do not have a dull moment. Like during the breeding season, like these males are working hard.
Good.
Another question that's more different topic is that Idaho and gobies, that sounds a little strange, right? So what's the story between?
How did that get?
There's no relationship between Idaho and these gobies. I mean, I started studying these gobies when I lived in Atlanta. Actually, I moved to Atlanta to study them just because my advisor was in Atlanta and that's where his lab was. So we can actually try, like they do relatively well in a lab environment. So I'm currently building a fish facility, so like an aquarium on campus where I can we'll have everything right so that we can house these fish there. And so I'll have to transport them and keep them there so that we can study them throughout the year. But then so that's kind of the relationship. And I got a job in Idaho, so I had to move to Idaho. Yeah, well, if there are no more questions, I guess that's where we wrap up today.
Thank you so much for being here tonight.
My pleasure. This was a lot of fun. Thank you. And I can actually hear your claps. Everyone muted. So, whenever we're giving talks, often people actually take away their video as well as audio and then the virtual clap. But thank you. I could actually hear your clap today.
That was very interesting.
Thanks. And I mean, it seemed like you were all following through. So that's great. Yeah.
Yeah, it's amazing. We're so much like animals, and yet people just fight that. And then you see this, and you go, "See, we're just like little fish, and they're just doing the same old thing that we're doing. We're just more sophisticated at it."
Maybe we're sophisticated, but we quarrel a lot more, and we hold grudges.
Yeah, we fight about a lot of things. Yeah,
these guys resolve things pretty quickly. Like
very very straightforward. Yeah. So no, that's really interesting.
Yeah, but yeah, I mean, them living in social groups is a lot like us living in those kinds of groups, and
people are always wanting to get other people. They're always wanting to make it difficult for someone else to do their thing. Up against, and people are always fighting and wanting their resources.
Their thing, and which is why this is where your tax dollars go to. So is to fund these kinds of research, and this kind of connection that you just made by yourself without me telling you is really important, because that's what that's what I need to tell the funding agencies.
Well, I mean, I would bet, and I don't know how anybody will determine this. I mean, I always say I wish I could do this kind of thing, but I will bet you that the females know which eggs are what, because it would make sense that they would say, "Oh, well, I need some protein, and I think I'm going to go for their protein. I'm just going to wipe them out. You know, but, I mean, it just seems that the territorial part of it would already be there. You know, just because humans kind of exhibit like, or lions. You know, the feline area, where they'll kill the cubs of a rival male, because it makes sense that they would say, well, I'm not going to let that, you know, happen. So it makes sense that the fish would maybe do that. They'd go, oh, I'm going to go over in this little part of the nest, and I'm just going to eat all of those because I don't like that bitch over there. You know, she laid her eggs while I'm getting rid of them.
Yeah, I mean, birds do that as siblicide. There are these like boobies, these birds that actually, and even eagles do that. They actually will feed the youngest, youngest hatch, like, you know, the most recent hatched chick to the oldest one.
Mm-hmm. It's a very vicious. Mother Nature is just harsh on these little guys. So
but that's basically it's all like very. This is how things are. If whoever is going to live and survive and be able to extract what they can.
That's
nature's interesting, especially with like the female praying mantises that will eat their mates like right after mating. So it's like
it's protein. Hey, insects have a lot of protein too, and that's really
it's all the protein.
It's all there. Yes, we should be eating more protein. Give up those carbs.
All right, thank you.
Thank you.
You're welcome. Thanks for joining in, and thanks, Chad, for the invite. I appreciate it a lot.
And you'll put it up, and we can post it, right, Chad? Is this one going to be?
Yeah, this one's fine to post.
Okay.
Yeah. My internet was terrible once again, so thank you to Lena. Said that she would record what she was doing.
Okay.
Yeah, I will send it to you.
All right.
Great.
Bye.
You'll send it to me, and I'll pass it out.
Bye, Dan. Bye.
Thank you. Bye.
Bye. Thanks.